And speaking of that, do you two online have any questions about anything while we're waiting?
No, sir.
Sounds good. All right. Well, if you think something, let me know. Good morning, Thorn Bar.
Good morning, sir.
5 minutes early, so you'll get a little bit of extra credit. .45 points as opposed to 2.4. Oh, so since I'm recording and I've got a small audience, I want to let you guys know about independent work. When obviously you need to include a source, but make sure your source is your actual source. Let me give you an example.
I got your email yesterday.
Okay, but I can show you what I mean. So let's say I want to say like, all right, I'm a fan of dogs. I'm gonna write a paper on, how long do they usually live? What is the average life expectancy? Oh, I can't see what I'm writing. Whatever. Expectancy of dogs. All right, so I typed that, right? And then, okay, that's an ad. I'm not going to do that. 10 to 13 years. So there's the answer. I mean, that's good enough, right? This is just independent work. So what I would do, though, if I'm writing 10 to 13 years, I'm click the actual link that says 10 to 13 years. And once it pulls up.
Good morning.
Good morning. That I would copy that, and that is the link I would put into my paper.
I got you.
As opposed to, all right, so I Googled it, and I don't want to copy this.
Right.
Because with a little bit of detective work, yes, I could figure that out and be like, okay, based on what that person said and based on their Google search. Because if somebody includes this is what I see. I see their Google search. I don't see what their source was. So
I think I fixed it.
okay, for anybody who's done that, make sure you can always go back and fix it. That's the beauty of independent work. It's fixable. Actually, everything is in this class. All right, so we have Huff is here. 3 minutes early, so a little bit of extra credit. 2.43 points. And Hamrick is online. Good morning, Hamrick. Let's see. Well, I guess I can X out of that now. It's just an example. I see Ray is online. And Johnson. All right, 7:59. We got a little bit less than a minute. I'll go ahead and start attendance, mostly for the sake of the recording. So Maxwell is not here yet. Same for Breeden. Same for Elias. Davis is online. Lloyd is online. Hamrick is online. O'Brien is here. Ray is online. Michael's not here yet. Dora Pena is not here yet. Sigmund is not here yet. Bayard, not yet. Johnson is online. Huffman is here. Michael is here. Online, that is. And we have Sullivan, not here. Hill is online, I think. Yep, Hill's online. Byrne is not here. Tyrus, not here. Lacey, not here. Thornabar is here. Fitzpatrick, is here. In person. I love it. Hamlet, not here. Huffman is here. Johnson is online. Bayard, nope. And then we had Cooper come in and Elias. Got you both. Anybody else that I'm missing? Haven't called. Okay. For those of you who are online, I'll remind you, pay attention, because just being here, as you know, won't give you the attendance points. Either there'll be questions later, or maybe I'll give you some instructions on an email you can send me. And speaking of which, pay attention to the emails. So I got an email. I haven't read it yet. Oh, it's a double part announcement. I have a lot of emails to catch up on. So if you sent me something and I haven't graded it or gotten back to you, I apologize. That's why. So don't feel like it's lost in the sauce. I just haven't gotten to it yet. But I did glance over and saw somebody sent me an email way past after I already sent or released the questions for Friday. And in that email, they said, hey, here's the keyword. I haven't responded to them yet. I don't even remember who it is, but that's too late. Once I post the questions, that means you were not there. You have to answer the questions once I posted the questions. Anyway, any questions about that?
Before you post the questions, you have to email you to say, hey.
Yeah, if that's the case. Yeah.
This is what you said.
Yeah. Usually.
Right.
Lacey's here. In person. All right. Anyway, that being said, I'm gonna keep doing this because it's only a week now. Independence due this time next week. It'll be too late, right? Because it's due like midnight of next Sunday. That midnight. So I want to remind you of a few things, even though I've said it before. I'm gonna keep saying it. That way, when you guys come in 2 weeks from now and say, "Hey, is there anything that you can do? I can do?" I'm like, "Yeah, you can go watch those videos I already posted." They tell you, "No, there's nothing you can do." Here's a little graph I like to make. This is the number of independent work submissions I get per day, starting from day 1 of class to the 25th of November or of October when it's due. It usually starts off pretty strong because there's some go-getters, right? And they're knocking them out. But then because they're go-getters, they're usually done like pretty early. So the daily submissions drop off because the go-getters are done, and the people who are waiting for some God unknown reason to the last day to do it, they're not doing anything. So I've got a small amount of submissions, and then what happens right about here is where we are now. It starts creeping up because we've got a week left. And then starting probably Friday, I'm going to get a ton of emails. Everyone's like, oh, I better do this. So I'm telling you right now, it's already too late in a way. Because I'm about to start getting inundated with emails, you might send me something today, and I might not be able to, because I'm going to have 1,500 emails, I might not get to it until next week. And I might get turned back and be like, I can't accept this because of this mistake, and this mistake. At which point it'll be too late, right? Because it'll be past the due date. But I promise you, I'm not going to do that on purpose. I promise I'm going to try to get it graded so I can get it back to you and you can correct your mistakes. But I can't guarantee you I'll be able to. That's why I've been trying to get you to do it all semester. Because at this point, you're getting to the point where it's going to be too late. I'm going to turn it back and say, oh, you need to fix this. I can't accept this. And then it'll be too late. So please get it done. Also, I want to remind you, I'm not doing this just for extra work. This is supposed to help your grade. Ironically, most people who needed the extra points because they're just not doing good in exams are also the people who are not doing good in independent work either. But it's there for that reason. If you know not a good exam taker, independent work is just free points. Write some papers. Bump your grade up because you know you're not a good exam taker. That's what it's there for. I'm trying to help you. Any questions about independent work? All right. And like I said, it's getting too late because of me being inundated with emails. But I'm always here for you. You can always visit during office hours. But look, I know you've said it a million times, but please clarify this again, and I'll be glad to do it. I'm not trying to make this hard. I'm trying to make it easy. And it was supposed to be enjoyable, but if you haven't written anything yet, then even if you find topics you like, there's nothing fun about writing, about 10 papers in a week, 10 pages in a week, excuse me. Anyway, let's get into it. We are now starting chapter 9, which is all about genetics. In my opinion, in today's situation, I would say this is the most pertinent chapter we're going to cover this semester because of the pandemic. So obviously there's a lot of information out there about the pandemic. How deadly is this? How deadly is this virus versus that virus? How communicable is it? What's the difference between the original virus and the Delta variant? How do vaccines work? Are the vaccines safe? Do masks work? All that's, this really won't help with masks. But to understand all that stuff, I'm gonna make sure you get these quotes here, do your own research. You need to have some background information about how genetics works, how viruses work, and that is this chapter. So again, in my opinion, this is probably the most relevant chapter you're gonna have this semester, at least in the middle of this pandemic. So let's start off with a little guessing game. Your picture always includes some pictures. Your textbook always includes some pictures at the beginning of the chapter that tries to illustrate why this chapter is relevant to your everyday life. In this case, why genetics matters. Obviously, I already told you the biggest thing is COVID-19, right? To fully understand that, you have to have some base information, and that'll be this chapter. But how about this? This lady right here, look at her hair color, look at her tattoos. Now, if she were to have a kid, do you think that kid would come out with that hair color?
No,
Why not?
because that's artificial.
Yeah, okay, right. Because that's probably, I assume that is correct too, right? That's probably not her natural hair color. Now, do you think that kid's going to come out with an arm sleeve of tattoos?
No.
Why not?
Because that's something that she added.
Right. Again, something that she added, something that's artificial. And what you're going to learn, and hopefully what you already know about genetics, is just because somebody has something doesn't mean doesn't mean you can pass it down. And that'll be a big thing. Obviously, tattoos and hair colors, it's known. But there's other things where the environment influences your body, and there's these changes that your body makes and your cells make because of the environmental influence. But that doesn't necessarily mean that those changes are going to be passed down to your offspring. How about this? Can anybody even tell what this is a picture of? You see here what this is? It's a little bit bright down there.
Looks like the father and his son, or his daughter.
Actually, a good guess. It's hard to tell with babies, right? But yeah, that is in fact a father with the daughter. Any idea why they would choose this picture for a chapter in genetics? What might they be trying to illustrate there?
Does she have the house?
I don't know. But it's funny you should mention that. Off the top of my head, I think we're done talking about defects other than maybe sickle cell disease. But that's a good guess, especially because it's really hard to see. I'll just go ahead and tell you. I don't want to waste a lot of time. So that's the father and the daughter. And your book points out, and we'll learn a little bit why later in the semester, in this chapter, that it is the father that determines whether or not the kid's going to be a male or female. And you should already know that. You already know what you need to know to understand that. You know that a male is XY and that a female is XX. And we're going to look at more Punnett squares in this chapter, and you'll understand, oh, yeah, that's why it's the father who determines it. So all the females in here, the reason you're a female is because your father, you'll see why, but it's because of your father. And then here's a picture. It's a family. So hopefully what you can see in this picture of people who are related is that there's similarities and there's differences. And what your book is getting at is, some things are inherited, some things are genetic, some things are strictly from the environment, and some are a mixture of both. Sometimes the environment does affect your genetics. That's something called epigenetics, and we'll talk about that in this chapter. So any questions about this? Obviously nothing on there will be on the exam. All right, let's move forward. Every chapter starts off with this biology and society, where they give you a little bit of a story of something that has something to do with this chapter. And it's usually something that they think you would be interested in. And as always, I'm going to skip it. But this one's about dogs. You can read about it if you want. A lot of people like dogs. So it talks about the genetics behind dogs, the artificial selection, like how did we get to all these different breeds? I will say that the current hypothesis, at least last time I read, and you can check this for independent work if you want, but I think the current hypothesis is there was one wolf-like ancestor, and from there it split into two different species, which is wolves and what we know as dogs. And so where all dogs came from, they used to all be something that looked a lot like a wolf. And then humans got involved, and we got Great Danes and teacup Chihuahuas and everything in between. That's what that chapter, or this portion of the chapter basically says, and you can read all about it. Okay. Woo, here we go. This is a very information thick chapter. This one's going to take a while, so I apologize in advance. But we're going to learn all about genetics and heredity. And I like the way your book does it. It almost introduces you to it as a story, because the person who brought us modern genetics, or genetics as we know it, his name was Mendel. So your book kind of presents it like, all right, here's what we knew before Mendel. Here's what Mendel figured out, and here's how he did it. So it's going to kind of be presented as a story. As I'm saying this, I want you to know, or as I'm going through this, I want you to know, this is not a history class. So I'm probably not going to ask you any direct questions about Mendel, like what was his name and where exactly did he study. But there will be pertinent information about his experiments, and I'll let you know which parts are important and which parts aren't. So any questions yet? All right. First of all, heredity, before we get into it. Can anybody tell me what heredity is? I mean, you all know, I'm sure, but it's a little bit harder when you're sitting in a biology class and someone that asks you to give the definition. How would you define heredity? Any guesses? Yes, sir. I like that. Past generations, right? Meaning I would assume that like all the genes you have in you came from past generations, mixtures of past generations. That is true. Yes, sir.
Inherited traits.
Inherited traits. Good. I like that because you also included, heredity, inherited. Those words go together. Anybody else?
It could be things dealing with your medical, like heart disease.
That's true, right?
Cancer.
Yeah, medical stuff. So you might be more predisposed to a certain disease because of your genetics. Yeah. Your book definition of heredity is the transmission of traits from one generation to the next. And if I remember correctly, the definition in the lab was a little bit different. And I don't remember what it said. Does anybody even remember what it said? Because I remember in the pre-lab, it said, what is the definition of heredity, I think? And then the next question was, what is the definition of genetics? But anyway, speaking of which, what is the definition of genetics? Any guesses?
DNA.
It definitely involves DNA, yes. I'll give you a hint. It really goes along with this first bullet point. Heredity is the transmission of traits from one generation to the next. Genetics, according to your textbook, is basically the scientific study of heredity. I might ask you those definitions on the exam. Most likely not, because this is a very, like I said, very information filled chapter. We're going to have a lot of information here. A lot more important things than just the definitions. As a matter of fact, as long as you understand how heredity works, you understand these different processes that are involved in genetics, then to me that it's a little bit less important than you remember the book definition. So again, like I said, the person that we call the father of genetics is this guy up here, Gregor Mendel. And your book is going to present this information in a story format. So here we go. He did his work in the 1860s. I'm not going to ask you that. What he argued was that parents pass their unique genes to offsprings. So people knew, obviously, even back then, we're like, all right, when this person gets together with that person, look at their kids. Obviously, they're passing something along. There must be something about the way they look and their height and their weight or whatever it is. There's obviously some influence that the parents are having on the appearance of their children. They knew that. But what Mendel was saying was there must have been something unique inside of their cells, right? And he was saying that, all right, they're passing some kind of thing. He didn't know what, but they're passing something along, a distinct thing. And I put genes on there because that's what we're calling genes. We know them today as genes. They didn't know that back then. So he didn't call them genes. He called them heritable traits. Not that you need to know that. But he was saying parents pass down the unique heritable traits to offspring. So like a blue-eyed person and a blue-eyed person, they're going to have blue-eyed kids. That was a heritable trait they passed on to their kid. Anyway, he also argued, again, I use the word genes here, but we didn't have that word back then. But he argued that these genes were responsible for the inherited traits. So again, blue-eyed person with a blue-eyed person has blue-eyed kids. There must have been a gene, not that he called it that, but there must have been something that was responsible for that. He also argued that genes retain their individual identities generation after generation, no matter how they're mixed up. And the eye thing is another good example of that. And I'll use myself as an example. I have blue eyes. My mom has brown eyes, and my dad has brown eyes. So even though both my parents had brown eyes, they had that gene for blue eyes hidden somewhere. And you'll learn where a little bit later. So they had the gene, they just weren't using it. Does that make sense? So no matter how it was masked up, it was still being passed down generation to generation. And then finally, he argued that genes retain their individual identities generation after. Oh, I already said that. I'm sorry. I got very little sleep last night. You'll need to forgive me. So any questions about what's on this slide? Most likely none of this will be on the exam, except, again, maybe the first two bullet points where I ask a definition. This third bullet point talking about Mendel, I'm definitely not going to ask you any of this. This is almost like an introduction. Like, hey, here's all the stuff we're about to talk about. Just an introduction. So any questions about that? All right. Independent work idea, if you want. This is really good because you can do a two-sentence independent work paper, or you can make it really long. But you can look up Gregor Mendel, find some information out about him. Anyway, moving on. Where did he do his work? He did his work in a place called Abbey Garden. You don't need to know that. But again, your book is presenting this information in a story format, so that's how they're doing it. In Abbey Garden. First of all, let's talk about what he studied. Because I think when most people think about genetics, unless they're a geneticist, they probably think about people, right? Because that's what's most relevant to us, arguably. But he studied garden peas. So the same old peas that you get from Kroger, right? Green peas. That's what he studied. Why did he study them? Well, first of all, because they were easy to grow, and because they came in many readily distinguishable varieties. And we'll talk about that second bullet point moving forward. But for now, obviously the easy to grow thing makes sense. So if we were to have an experiment throughout the semester, I would probably use something like a pea plant, and we would start at the beginning, and then by the end of the semester, we could, look at our results. Maybe like how do plants do when you give them warm water instead of cold water, right? We could have had pea plants. Half of them could have gotten warm water every day. The other half could have gotten cold water. And then by the end of the semester, we could have seen which one grew the most, right? We couldn't do that with a redwood, right? Those big trees that are hundreds of feet tall. First of all, it would just take too long to grow. Second of all, we couldn't fit it in this room, right? So peas were a good option. Not that I'm going to ask you that, but there you go. Now, here's some very important things you do need to know. And it's easy to mix them up, but it's important that you know them so you know what I'm talking about when I use the words in this chapter. The first one is a character and a trait. These words are different, but they're related. A character is a heritable feature that varies among individuals. And I'm definitely not going to ask you the definition. I'm just saying you need to understand this word when I use it. So, for example, hair color is a character. Eye color is a character. Skin color is a character. Height is a character. Dimpled chin, freckles, free hanging earlobes, bent little fingers. Those are all characters. And that brings us to something called a trait, which is a variant of a character. So if the character is eye color, then the trait could be blue, could be brown, could be hazel, could be green, right? Could be red if there's if you're albino, or it could be any other color if you're some other animal. Hair, again, the hair color is a character. Brown is a trait. Black is a trait. Blonde is a trait. Red is a trait. So you guys see the definition or the relationship then, right, between character and trait. And again, I'm not going to ask you that. It's just that I'm going to use those words a lot. You need to understand the difference between the two and understand the relationship between the two. So anyway, going back to the pea plants here that he studied, each character that he studied had two distinct traits. So for example, if he was using humans instead of peas, skin color wouldn't work, right? Because it's not like everybody's either this color or that color, right? There's a beautiful spectrum of skin color in humans. Even eyes, right? There's even blue eyes are not all blue, and even brown eyes are not all brown. There's all this in between. So he picked things that had it's either this or that, and we'll talk about that moving forward. But hopefully that makes sense, because if you're trying to study something, it's like, did this get passed on or did that get passed on? It helps if it's this or that. Height is another example of what would be bad, right? Because it's not like humans are either short or tall, right? There's everything in between. So anyway, let's talk about these pea plants again. An important advantage of the pea plants is that he could strictly control the reproduction, which is good. Because if you're trying to say, all right, what happens if we mate this with this? What's going to be the results? To be able to do that, you have to control it, right? You can't have accidental breedings happening. You need to control what is mating with what. You do not need to know any of this stuff. This is for your own knowledge, just so you understand how it worked. But I will not be asking this on the exam. How did he control it? Well, I'm gonna skip this and I'll just show you the picture because again, I'm not gonna ask you anything about it. These are the reproductive organs of the plant. So you can see here's the petal that encloses all the goodies, if you will. Here's the stamen. All these little things right here are stamen. Those are the things that make the pollen, which is where sperm comes from. So again, like when you go outside in spring and there's pollen all over the place, that's essentially plant sperm, and that's where it comes from, assuming it's from an angiosperm. Anyway, that's the thing that makes a sperm. This thing right here, the carpel, this is where the eggs come from. So what happens is the sperm and go in down through there, and they fertilize the eggs. So this is where you really need to think. Take a step back from what you know about sex. And remember that we're talking about plants, not humans, right? So if humans want to have children, you need a male, you need a female. Here, these things can reproduce with themselves, especially if the petal's closed and they're not even exposed. Like, yeah, they're putting out sperm, they're putting out pollen, but if the petals are closed around it, that pollen's not going anywhere other than right back into its own ovaries, to its own eggs, right? That might seem like a disadvantage because if you're trying to control what is mated with what, then obviously you don't want these things mating with themselves either. So what did he do? Well, I'm gonna skip this too. It's better to show you the drawing. First of all, he cut off the male parts, right? And then he took the sperm from one plant and put it on, made sure he fertilized a different egg with it. So he was able to control it that way. Again, none of this is going to be on the exam. The point here is that he was able to control what mated with what, and that's how he do it. All right, excuse me, that's how he did it. Peeled off the petals, exposed the sex organs, and cut off the male parts. Can't do that with humans. It's very unethical. Anyway, what happens when you fertilize a pea flower? Eventually, that turns into peas themselves, and what we know as peas, that's what it is, right? It's little babies, little baby pea plants. That's what seeds are, little baby plants. So a walnut is a little baby, walnut tree. Peas, those are little baby pea plants. Obviously, then, you probably know them as seeds, but what you can do with those peas, you can plant them, and you have a whole new plant. Here's some other stuff you're going to need to know, and the next slide I'm going to show you puts this in writing. But you're going to need to know the difference between the pea generation, and that's easy to remember because it's in the name, right? Pea, it's going to mean parental, and then the offspring, which is the F1. There's also something called the F2, but we'll talk about that here in a second. In my opinion, as long as you remember that P is the parental generation, then the rest of it should be pretty intuitive. So how did he do this? First thing he did was create purebred varieties of plants. And you do need to know this, at least for the study guide, maybe for the exam. What is a purebred variety of a plant? A purebred self-fertilizes to produce offspring that are identical to the parent. In the context of what we're saying here, what I mean is, if we go farther back to what I said, remember I said genes, even when they're masked and you don't see them, they still maintain their identity, right? So what we're saying here is that he bred and bred and bred and bred and bred to make sure all those other genes were out. Or as we know from the lab, the proper word is alleles. So again, using my parents as an example, yes, they both had brown eyes and I ended up with blue because that gene was hidden. Now, if he was doing an experiment with humans, what he would do was breed my parents and then look at all the kids and find the ones that had brown eyes and then breed them and then find their offspring that had brown eyes and then breed him. That's obviously horrible with humans, but that's what he did, right? He kept doing it until he was sure that any kind of genes, any kind of variation for any other trait was not there. And once he knew he had purebred plants, then he crossed these two different purebred varieties to come up with something called a hybrid. And yes, you need to know what that is too. Not only do you possibly need to know it for the exam and for the study guide, but because it's a word I'm going to use a lot. So what is a hybrid in this context? That is the offspring of two different purebred varieties. And I'll give you some examples later, but for now, I'll go ahead and give you a spoiler alert. One of the things he looked at, one of the characters he looked at was flower color. And it either came in two traits, purple or white. So first he created the purebreds. He had those purple plants and he kept crossing them, like self-crossing them until he knew that there were no more genes for white. Same for the white. He kept crossing those and crossing those and crossing those until he knew he got rid of the genes for purple. And then he took those purebred white ones and the purebred purple ones and he crossed them, and that was called a hybrid. And here's some more words you need to know that are involved with hybrid, and you do need to know this for the exam, and you do need to know it because I'm going to use it. The first word. So when we say that we cross these purebred varieties to get the hybrid, what do we call that action? What is the name of that verb when we cross them? That is called a genetic cross. And yeah, now that I'm looking at it, that most likely will not be on exam, but I will use that term, so you need to know what I'm saying when I use it. So when we cross two things, it's a genetic cross. Now, what are the two things that we're crossing? I wish I was really good at English. We could do a sentence and diagram it. Like, here's the subject. But anyway, what are we crossing? Well, these two purebred plants, those are the P generation. And you do need to know that. We're going to use that a lot. So we start off with the P generation, this male with that female. Their offspring are called the F1 generation. And then if you were to cross the F1 generation and look at their offspring, that would be called the F2 generation. So again, using my parents as an example, and I hate to do this because it's humans and it's really disgusting when you think about it in terms of humans and not plants. But if my mom and my dad, both with brown eyes, but both with hidden genes for blue eyes, if they had kids, just a bunch of kids, you would look for the one. If you try to create more brown eyes, you look for the kids from the F1 generation that have brown eyes. I'll just put BR, brown, and you're going to mate those kids with the other kids that also have brown eyes, and then those kids would be the F2 generation. So if you were thinking about this as humans, you could almost think of like I don't know, that's your generation. And then above that would be your parents. I'm going to abbreviate because it's really hard to do that. Then above that would be your grandparents. Any questions about that? I also point out that this is a very simplified version. You are learning a very, very simplified version. Because this is mostly what he did. If we're talking about genetics in general, excuse me, especially with plants, because agriculture is obviously a big thing. In real life, it's a little bit more complicated than this. Obviously, we go farther than the F1 generation and the F2, right? We keep going. And also, sometimes we might cross this F2 generation and cross it, go back to a plant from the P generation or from the F1 generation, right? It gets really crazy. But for the sake of this class and for the sake of understanding what Mendel did, just know that you have the P generation, then the F1, then the F2. Are there any questions? All right, for the exam, I'm not going to try to trick you. So if you know, F1 and F2, that's easy to remember because it's 1 and then 2. And with this multiple choice exam, it's not like there's going to be, your choice is going to be, is it F1 or is it P1 or is it, O1? Nothing like that, right? F1, F2, those are the only options you're going to get. I'm not going to try to trick you with some letter that might work other than F. So no questions about this slide? All right, that was the easy stuff. Let's jump into the little bit more difficult stuff. Let's talk about Mendel's law of segregation. This is a big one. There'll be probably a few questions about this. And obviously, because it's a big one, we're going to talk a lot about it. Mendel's law of segregation. First of all, this is just the intro, nothing to write down here. Obviously, Mendel performed experiments. That's what we're talking about here. In these experiments, like we've already said, he tracks the inheritance of characters. In the example I've already given, and the example your book is giving is flower color, right? Flower color is the character. And remember, the trait would be the different varieties. So in this case, it'd be purple or white. So this is just a reminder of what I've already told you. The results of these experiments that he did led him to formulate several hypotheses about inheritance. And before I give you those hypotheses, I'm going to give you a picture that illustrates what he did. Here we go. This is a whole list of the different characters he looked at. Flower color was a character. The different traits were purple and white. Flower position was a character. The different traits were axial or terminal. We're not going to go through all these. You definitely don't need to know them all. These are all examples. So definitely will not be a question where I say, hey, which characters did Mendel work with? That's not important. It's just letting you know that he worked, even though we're only going to talk about 1 or 2 characters, he worked with a lot of them. That's, I guess, the point here. And again, they come in only 2 different varieties. They're either dominant or recessive. And we've already talked about that lab, and we'll talk about that more here in this chapter. But there were no in between, right? There were no light colored purple, right? They're either purple or they're white. The seeds were either yellow or green. There were no mixes of the two. Yeah, and they were either tall or short. They weren't in between. Any questions about this? All right. You already know what a genetic cross is. Now, here's a subcategory of a genetic cross. It is called a monohybrid cross. And oddly enough, you already know about monohybrid crosses. I just never called it that. So the things you did in lab last week, where you had to do those Punnett squares with, when you were looking at your alleles for chin dimple versus your mate's alleles for chin dimples, and what would the offspring be? That was a monohybrid cross. So what is a monohybrid cross? It is a cross between, well, in this case, purebred parents that differ in only one character. So again, if Mendel's looking at flower color, he doesn't care about the rest of it. Pea color, who cares? Height, who cares? Doesn't matter. He's only looking at the flower color. Is it purple or is it white? So any questions about what a monohybrid cross is? And mono being the key. And you know what? I guess I shouldn't. Let me backtrack. So what you did is not a perfect example of a monohybrid cross, because first of all, it was just hypothetical. And second, you're not necessarily purebred, right? So especially if you have brown eyes, you might have genes for blue eyes in you that's just not being shown. So you're not necessarily a purebred, in which case this wouldn't be a monohybrid cross because you're not purebred. But anyway, moving forward, here's an illustration of what we're talking about. You mix the purple flower with the white flower. What color do you think? And you've already done something like this with these Punnett squares. So without doing a Punnett square in your mind, what do you think the F1 generation looks like? all their offspring. Do you think they're all purple? Do you think they're all white? Do you think they're half purple? Do you think they're half white? Do you think there's some other ratio? Remember those ratios you did, like 4 or 2 to 2 or 3 to 1? What do you think the ratio was?
It's probably 1 to 1.
I like what you're saying. In some situations it would be. But the key here is that they're purebred, meaning, and then again, I drew this up here. These are dominant. The purples are dominant. The whites are recessive. So that means this thing is homozygous dominant, and this thing over here on the right is homozygous recessive. And your book doesn't point this out because they're going to introduce you to that later. But I'm saying it because you already know about it from the lab. But what happens when you cross them? All the F1 generation are purple. And that's a pretty big thing. And you guys are probably pretty used to it because you've seen something like this before and you've learned a little bit about genetics even already. But at the time, that was a pretty big deal. And to be clear, this is just a simplification of what he did. It's not like he took one purple flower plant and one white flower plant and crossed them and looked at the one offspring, right? He took hundreds of purple plants, hundreds of white plants, had thousands of offspring, and this was always the outcome. 100% of the F1 generation was always purple. So then you have to ask, well, what happened to the white gene? Is it gone? Or as we know, allele, but at the time it didn't. What happened to the heritable trait that was responsible for the white flowers? He didn't know that. So what did he do? And your book doesn't show this, so I will. He took another F1 generation and crossed it with this. So my little flower. That's horrible. All right, there's a flower. So he crossed another purple flower with another purple flower, both of which from the F1 generation. And then that led to the F2 generation. And what do you think the F2 generation looked like? You think they were all purple? Do you think they were all white? Do you think they were 50-50? Any guesses?
50-50.
That would be a good guess too. And I'm glad you think that because that's exactly what I would have thought. That is exactly what I thought when I was a student. But it's not 50-50. And if you really thought about it, and if you had the chance to draw it out, you would probably come to the correct conclusion, which is that it was 3 to 1. And again, it's not perfect, but he did thousands of plants. And generally speaking, every time he did this, it was about 75% of them were purple and about 25% of them were white. Any questions about that? All right, pretty simple so far, right? So what he determined from this was that the gene for the white flower didn't disappear, right? So he looked at the F1 generation, always 100% of the time, they were purple. So that really made him think because back then, no one knew anything about this. So again, scientific process, that was his observation. They're all purple. Question, where did the white gene disappear? Hypothesis, no, it did not. How did he test this hypothesis? Well, he crossed the F1 generation. He crossed this purple flower with this purple flower, which you would think would end up with a purple flower, but as we know now, it was only 75% purple and 25% white. So there we go. Any questions about that? All right, let's move on. So based off of that very simple experiment, or experiments, because again, he did that thousands of times, based on those experiments, he came up with 4 hypotheses that you need to know. I'm going to go really slow because somebody had to step out. I'm going to make sure they get some of this. From these results and other results, he did other experiments other than this one that I showed you, but they're all pretty much the same, looking for the same thing. Anyway, these 4 hypotheses. Number 1, there are alternative versions of genes that account for variations in inherited characters. Again, these as alleles because we've already had this in lab. But that's a big thing too. So what we're saying here is there's not necessarily a gene for purple and a gene for white, going back to the flower color. What we're saying here is that there's one gene for flower color. And he figured out through these experiments that there's only one gene. There's not 2 separate genes. There's one gene, but there's just different versions of that same gene. That's the first hypothesis. In my opinion, that's the least important hypothesis as far as the exam is concerned, but there it is. There's the first hypothesis. Like I already said, and like you already know from the lab, what do we call these different versions of the genes? We call them alleles. Very important concept, right? You've got one gene, but you have different alleles. And I also point this out, even though we'll talk about it later. Keep in mind, what we're talking about right now is a very simplified version. Most things don't have one gene with two alleles. But obviously, we want to start off easy. So we talk about these things that have one gene and two alleles. Even the stuff we learned in lab, if you want to look it up for independent work, not all of it was as simple as one gene and two alleles. But they are very commonly used in genetics labs because they're close enough to one gene, two alleles, that we use them in basic biology courses. But yeah, look them up if you want. Chin dimple, freckles, eye color, tongue roll, hitchhiker's thumb, crooked finger, whatever it is. So that's the first hypothesis. Any questions about his first hypothesis? Pretty simple, right? Second hypothesis. For each inherited character, so remember, character is the big thing. That's like saying flower color is the character. Purple is the trait, white is the trait. For each inherited character, an organism inherits two alleles, one from each parent. And this is stuff you already know too, right? So far, this is like a review of what you already know. But are there any questions so far? All right, so if you have two alleles for each gene that you got from your parents, let's say you have brown eyes and you got both of your alleles from your parents, right? Your mom gave you a brown allele and your dad gave you a brown allele. How would you describe your genotype? Are you homozygous dominant? Let's make it easier. Are you homozygous or are you heterozygous? And again, you got a brown from your mom, you got a brown from your dad. What are you, homozygous or heterozygous? Yes, homozygous, right? Because you have two of the same. You need to know this. We talked about it in the lab. I'm definitely going to talk about it a lot throughout this chapter. It'll definitely be on the study guide. It will definitely be on the exam. Homozygous, two identical alleles. Of course, in the case of eyes from the lab, in that situation, you would be homozygous dominant if you had two brown alleles. You'd be homozygous recessive if you had two blue alleles. All right, so again, that's what a homozygous genotype is. What is heterozygous? Different, right? It's that simple. Homo, same. Hetero, different. Organisms that have two different alleles. So both my parents had to be heterozygotes for eye color. Because if they were, either one of them were homozygous dominant, first of all, they both had brown, right? So we know they were either homozygous dominant or heterozygous because we know they had at least one brown allele. Now, for them to pass on the blue allele, that means we know they were not heterozygous. They have to be heterozygous. They couldn't have been homozygous dominant because they needed at least one blue allele to pass down to the next generation. So again, this should all be a review because luckily for us, we already talked about this in lab. All right, here's a little picture. I didn't make this. I just found it, stumbled upon it on the internet. Heterozygotes, just a little uneven. Anyway, number 3, his third hypothesis. If two alleles were different, of course, he didn't call them alleles. He didn't know that word yet. That means one determines the appearance, and that is dominant, and the other one has no noticeable effect, and that is recessive. And you already know this from the lab, right? Dominant and recessive. I see people taking notes. That's why I'm pausing. So if you have the dominant, that one's dominant, right? They call it dominant because it's dominant to the recessive. So it doesn't matter what the recessive allele is. If you have the dominant allele, that's what you're going to show. Any questions about that? All right, let me ask you, if you were to write a dominant allele versus a recessive allele, how would you write it? Uppercase or lowercase for dominant? Uppercase, you already know that from lab. Therefore, by process of elimination, recessive is lowercase. You already know this. You've already done it in lab. Any questions about this? All right, if you're online, send me an email for attendance. And today's keyword will be tissue. I don't know. Send me an email, say I'm writing for attendance. Today's keyword is tissue. And obviously, if I post questions, it's too late. So you need to get on it. Anyway, moving forward. Any questions about his third hypothesis? All right, his fourth and final hypothesis, at least that we talk about. A sperm or egg carries only one allele. And you already know that from chapter 8. You know that from meiosis. When you produce a sperm or an egg, all your chromosomes split, right? And then your sister chromatids split. So you only have one allele. So in your cells right now, in your somatic cells, for example, using eye color, you have two alleles. You have an allele that you got from your mom. You have an allele that you got from your dad. But then when they split, when you make sperm or egg, and you were to look at all your sperm and all your egg, assuming they all survived, 50% of them would have that allele that you got from your mom. 50% of them would have the allele that you got from your dad, because they separate, right? Or a better word for it is segregate. And that brings us to this. Everything you just learned right now from what I told you and what you learned in chapter 8, there's a word for it. And it's called the law of segregation. And it's just simply saying when you make sperm or egg, every single allele pair that you have separates. You only have one allele for each character, for each gene. And remember from the experiment, just because something is dominant doesn't mean it's more likely, right? So if you were to look at all your sperm, if you're heterozygote for anything and look at all your sperm, 50% of them would be have the dominant allele, 50% of them would have the recessive allele. It's not dominant because like you have more of the dominant allele than you do the recessive, right? It's still 50-50. It just so happens when you put the two together, dominant always wins. So dominant has nothing to do with how many you get, because that is an option on the study guide. It says that's something about the law of segregation. And remember, you only have one allele per gamete, per egg, per sperm, however you want to say it. Only one allele. Here's what we're saying. Here's a picture of what we're talking about. There's your F1 there. This is a picture you've already seen, except now your book threw in the letters, right? So now here we go. Purebred purple, homozygous dominant. Purebred white, homozygous recessive. What happens when you cross them? You get 100% of them, 100% of this F1 generation is purple. But we know, if you were to do the Punnett square, you would know that they are all heterozygotes, right? Now, what happens when you cross a heterozygote and a heterozygote? That's right here, right? Heterozygote and heterozygote. You get, for a phenotype, 3-to-1, right? 3 of them are going to be dominant. 1 of them is going to be recessive. 3-to-1 phenotype. For the genotype, you're going to have 1 homozygous dominant to 2 heterozygotes to 1 homozygous recessive. These are things you definitely need to know for the exam. One of the questions is going to be, when you see a 3-to-1 phenotypic ratio, what should you associate with that? And the answer would be a monohybrid cross. We're still talking about monohybrid crosses here. Later, I'll introduce you to something called a dihybrid cross, and that will have different ratios. So I'm telling you right now, you need to recognize that 3-to-1 ratio as a monohybrid cross, and that 1 to 2-to-1 ratio is a monohybrid cross. Any questions about that? All right, this next slide is going to be really easy because we've already talked about it in lab. The next slide I'm going to show you tells you what a Punnett square is, but you guys already know what a Punnett square is. And based on the labs, you all know how to do a Punnett square, but also based on the labs that I've seen, you do need a little help interpreting the Punnett squares, and I'm here to help. What is a Punnett square? It highlights the 4 possible combination of gametes, right? 1, 2, 3, 4. You can see that at the bottom. There's 4 of them. 4 possible offspring. Each square represents an equally probable product of fertilization. We've already showed you the picture. So when we come back on Wednesday, we will talk about alleles and homologous chromosomes. Again, it'll be almost like a review from what you learned in chapter 8, except we're looking at it under the context of genetics. So a little bit of a review with some new words thrown in. Any questions? All right, I'll see you guys on Wednesday or during office hours. And I'm still offering a little bit of independent work just to show up at office hours, online or in person. A lot of you should have questions based on the grades. So I don't know. If I were you, I would show up online. I'm like, hey, can you explain this? Gaga. Then yes, I will, and you'll get some extra points. Anyway, that's it. You guys have a good Monday.